So hello to all. Now we'll talk about glycogenolysis. So first of all, understand what is glycogenolysis. It is the breakdown of glycogen into glucose. So when it occurs, whenever there is less or whenever decreased blood glucose level is there, at that time glycogenolysis occurs. Most common case is your fasting. So let us understand the cycle in very simplest manner. So you can see here glycogen is converted into glucose 1-phosphate. Glucose 1-phosphate is converted into glucose 6-phosphate, which is converted into glucose. So two enzymes are more important. There is a glycogen phosphorylase, which is known as a key limiting enzyme of the glycogenolysis cycle. It begins or it marks the beginning of your glycogen conversion. And we have G6 phosphatase, which will convert your glucose 6-phosphate into glucose. So remember, most often we can see such glycogenolysis in liver, kidney and muscle. But liver, kidney will contribute to the blood glucose. But muscle, we don't have G6 phosphatase. So glucose 6-phosphate will not convert it into glucose in muscle. So what happened to glucose 6-phosphate? It will undergo glycolysis and it will convert it into the pyruvate and lactic acid. Fine. So let us understand what is minus and plus. So remember, your ATP, CMP and calcium will activate your glycogen phosphorylase, which will help in this glycogen breakage into glucose. But if insulin is there, it will not allow the glycogen phosphorylase to become active. Now let us understand few clinical points. So as we know that the glycogenolysis mainly occur in the liver and is stimulated by the hormones glucagon and epinephrine. So whenever the blood glucose level decreases, like in fasting, there is increasing glucagon secretion from the pancreas. So if glucagon secretion will increase, automatically insulin secretion will decrease. Then what is the main function of the insulin? It's to store glucose in the form of glycogen. So opposite thing will occur. So whatever the glucagon is there, it will travel to liver and it will stimulate glycogenolysis. Now let us talk about few glycogen storage diseases. So very simple shortcut to remember here, that is Valerius President called and made her PM. So V stand for von Gogh disease. Here we can see the deficiency of glucose 6-phosphate. P stand for Pompe. So here we have the lysosomal alpha-1 glycoxidase. C stand for Cori. Here the deficiency of D branching enzyme is there. A stand for Anderson. Here branching enzyme is absent. M stand for McArdle, where there is absence of muscle glycogen phosphorylase. H stand for horse, where there is hepatic glycogen phosphorylase is absent. So you can remember the glycogen storage disease. One more important thing to shortcut. So G stand for GERK, G glucose. We can compare your POMP OM to the lysosomal. C Cori, D branching. CD you can remember. Here you can remember A, B. Then here McArdle, same muscle, horse stand for the hepatic. So these are few important glycogen storage diseases.